Hello, everyone. My name is Sonya Sar, and I will be presenting an analysis of the therapeutic strategies used to treat ALS exacerbated by TDP-43 aggregation. Amyotrophic lateral sclerosis, or ALS, is a neurodegenerative disorder that affects the function of motor neurons and, in most cases, leads to neuronal cell death. As neurons slowly deteriorate, they are unable to pass messages from the central nervous system, which includes the brain and spinal cord, to the peripheral nervous system, which includes the surrounding nerves. Doctors categorize ALS into two subdisorders: familial ALS, which is caused by hereditary genetic mutations, and sporadic ALS, which has no common underlying cause. Some symptoms of ALS include muscle weakness, spasms, involuntary movements, and paralysis, and the disorder is diagnosed by analyzing these symptoms. There are three commonly used medications for ALS: riluzole, edaravone, and sodium phenylbutyrate taurursodiol. Riluzole, the most commonly used medication for ALS, blocks the release of excess glutamate through oral or intravenous administration. Glutamate is a neurotransmitter, and its buildup in the synaptic cleft, which is the space between two neighboring neurons, can cause the glutamate receptors overactivation. However, riluzole has been seen to not only block glutamate receptors, but also acetylcholine receptors in neuromuscular junctions, which would prevent signals from being sent to muscles and exacerbate muscle degeneration in some patients. Since 1997, studies have linked riluzole to higher levels of liver enzyme concentrations, sometimes 2 to 5 times over the normal limit. Though riluzole increases a patient's life expectancy by 2 to 3 months, it induces at least one adverse effect in up to 50.3% of patients. The risks that come with the treatment in exchange for life expectancy means that it can't be labeled as the most effective treatment pathway. The second type of medication is intravenous and oral edaravone, and it focuses on providing cytoprotection to shield neurons from excess glutamate by detoxifying the surrounding reactive oxygen species. ROS cause damage to basic molecules like DNA, proteins, and lipids. One set of trials was done on ALS patients to test IV edaravone's effectiveness, and the disease progression was slowed by 30% after 24 weeks of treatment. However, this only proved short-term benefits to a specific category of patients. Another trial testing oral edaravone effectiveness showed positive outcomes, but only for patients with non-severe degeneration. However, patients approximately will pay $1.957 million in exchange for these benefits for each year of increased life expectancy gained. And this is not feasible for every single patient. The third medication type is sodium phenylbutyrate taurursodiol, which targets the histone deacetylase inhibitor by remodeling chromatin. The primary evidence for the effectiveness of sodium phenylbutyrate taurursodiol comes from the Centaur trial, and it decreased symptoms of ALS in patients whose symptoms began less than 18 months before. Overall, these targeted causes are not shared by a majority of the patients, so these treatments cannot help a majority of them. But what role does TDP-43 play in this disorder? TDP-43 is a protein that makes up a common aggregation found in 97% of ALS patients. It is not a direct cause of neurodegeneration, but it can progress the disorder's symptoms. Its functions are regulation of mRNA splicing and translation and nucleocytoplasmic transport, and aggregation in cytoplasms prevent this. This can be caused by mutations in the glycine-rich regions of the protein and the autoregulation pathway of the protein's production. However, what researchers debate on is whether the aggregation's consequences are caused by loss of function or gain of function mutations. The loss of function hypothesis states that the mutant TDP-43 loses some of its primary functions. Previous models of loss of function knocked down the TAR-DBP gene in both non-mammalian and mammalian species. They all reported that subjects developed neurodegenerative properties. Partial knockdown led to 601 mRNA strands changing and 965 altered splicing reactions. Silencing the TDP ortholog in C. elegans and D. melanogasters led to defects in growth, locomotion, and fertility. Lastly, simulating TDP-43 knockdown showed that proteins linked to ALS's symptoms reacted very similarly, proving a connection. The gain of function hypothesis, however, is derived from the idea that the TDP-43 gene is overexpressed, which leads to the protein garnering additional properties. Initially, it was thought that it led to novel toxic properties, and this was thought to be proven in vivo experiments, where protein aggregation caused by overexpression led to toxic properties. However, the expression of a mutant version of TDP43 that affected localization showed that it was not novel toxicity, but mislocalization impacting functions. This study also proved that this hypothesis is dependent on functions related to RNA processing because the overexpression's toxicity depends on RNA binding domains. One toxic mechanism is the blockage of intracellular transport related to RNA processing, and this linked the TDP43 aggregation to ALS. Overall, both mechanisms have equal proof to support it, so it is most effective to consider the possibility of TDP43 having pleiotropic properties expressed, gains of toxicity of TDP43, and the loss of its original primary functions. In order to determine more effective therapeutic pathways, researchers have been looking into three fields: gene therapy, antisense oligonucleotides, and small molecule/protein drugs. Gene therapy is a form of treatment that aims to manipulate or alter the expression of a gene in order to reverse the effects of diseases. It could be administered in two different ways: ex vivo and in vivo. Ex vivo targets includes target cells being removed from target tissue and edited before being inserted again. In vivo includes the insertion of the therapeutic gene into the patient's tissue or bloodstream. The gene can be encased in viral, bacterial, or lipid vectors, but ALS research uses bacterial vectors known as adenovirus associated vectors. One symptom of ALS, the loss of the integrity of cortical motor neurons, was analyzed by targeting the enzyme ubiquitin C terminal hydrolase L1, or UCHL1, that maintains a level of free ubiquitin. Free ubiquitin allows for normal nervous system development, and its deterioration leads to ALS symptoms. By using AAV-mediated retrograde transduction, researchers reduced the extensive integrity loss caused by mutated human TDP43. Another study focused on the human frameshift mutation by increasing its production since it expresses protective effects in rat paralysis models. Introducing UPF1 in AAV delivery, researchers showed that the models gained forelimb motor function. The third chosen study, however, chose to analyze how effective the method of delivery was for gene therapy in relation to aggregation. They tested AAV9's ability to deliver genetic material, but the promoter of the gene was found in the liver 4 weeks after first results. Therefore, they also tested a variant of AAV called AAV PHP.B that was engineered to enhance efficiency and carry TDP43's gene. Wide-scale IV delivery for TDP43 expression using this variant showed that it was more effective. However, it is hard to implement since it is more expensive. Gene therapies were used by researchers to analyze the effectiveness of targeting TDP43 and its relation with other proteins, including SARM1, which increases degeneration in neurons. One study used TDP43 to manipulate levels of SARM1 through STMN2, since TDP43 mediates STMN2's mRNA splicing, and overexpresses NMNAT2, a protein that can protect axons if overexpressed and inhibit the function of SARM1. And this was in order to reduce motor neuropathy. Genetic therapies have both positive and negative consequences. Its benefits include more successful treatment because it is specific to certain genetic mutations and applicable to a key group of patients. It also targets the causes of ALS symptoms and is supported by multiple trials of treatment in multiple animal models. Lastly, it does not have to be repeated in numerous times since it can have permanent effects. Its drawbacks include not being applicable to general population, making it more expensive and inefficient to create since most efforts and funds are spent in deducing the problem gene. Lastly, genetic mutations are only found in 10% of patients with ALS. The next potential therapeutic are antisense oligonucleotides, which are single-stranded DNA strands designed to complement sections of target mRNA and silence its expression. And though it is a form of genetic therapy, I decided to analyze it separately since it has some important characteristics, and research in ALS has focused on it specifically. The first study analyzed the effectiveness of the administration of ASOs by analyzing the administration of anti-SOD1 ASOs through direct delivery. It was more effective than other gene therapies mentioned, since subpule injections of the ASO showed that ALS's progression is prevented or stopped depending on what point in disease progression it was administered. Another study demonstrated how ASOs are used to target the levels of TDP43 aggregation without reducing levels of STMN2. Previously, when toxic TDP43 cytoplasmic aggregations were targeted using ASOs, STMN2 levels decreased, which is a symptom in ALS. This study therefore targeted the levels of TDP43 by blocking STMN2 cryptic splicing using an ASO. Targeting the cryptic exon using ASOs can increase STMN2 expression and reduction of either TDP43 or STMN2 mRNA and induced pluripotent stem cells. And this can restore axonal regeneration as seen in another study. Lastly, it is possible to increase the effectiveness of treatment against TDP43 by using ASOs packaged to target multiple cryptic exons simultaneously. What are the benefits and drawbacks of antisense oligonucleotides? The benefits include silencing genes and restoring protein function and expression while reducing the expression of toxic proteins. It is also helpful in the sense that it can be used to target genes individually and for combination therapies. The drawbacks of this method include continuous dosing to maintain response, which can be undesirable for patients since the delivery method is very invasive. Cellular uptake is also harder to control, but the introduction of liposomal delivery, which increases ASO stability in bodily fluids and facilitates its distribution, has increased the success of cellular uptake. Lastly, it maintains an accurate delivery of oligonucleotides to the action site, which increases the effectiveness of treatment. The last type of therapeutic strategy is small molecules and protein drugs. Small molecules are molecules found in drugs that are designed to interact with target proteins in specific ways, while protein drugs are used to recognize and target specific proteins in cells. One study proposes the use of calcium channel agonists, which are substances that bind to channel receptors to protect against neuromuscular dysfunction that is seen by many ALS patients. When an ALS mutation was expressed in zebrafish larvae, the characteristics of its motor function prior to treatment and after treatment using the calcium channel activists FPL 64176 or BK 8644 were stabilized after treatment. Another study analyzed the use of methylene blue, a medication to treat methemoglobinemia, and dimebon, an antihistamine, on TDP43 aggregation. These two small molecule medications have been successful in phase 2 Alzheimer clinical trials, and their combined use reduced the TDP43 aggregations in neuroblastoma cell lines by 80%. Patients with both FALS, and SALS caused by TDP43 aggregation have impaired autophagosome formation and the accumulation of glutamate receptors. One study showed that an anticoagulation deficient form of activated protein C, which is a glycoprotein that controls blood clotting, can reduce the presence of these defects and induce motor neuron models. Lastly, one study showed that when delivering using an adenovirus vector, derived single-chain antibodies were able to reduce toxic aggregation, neuroinflammation, motor defects, and cognitive impairment in mouse models. Like the other two treatment mechanisms, small molecules and protein drugs have both benefits and drawbacks. It has a more broad effectiveness for both FALS and SALS models, and there's a greater push for its use for two main reasons. It's easier to administer because small molecules can be provided in the form of medication and protein drugs through IV injections. Additionally, small molecules are an accepted treatment model, so it'll be easier to convince patients to use it. Some drawbacks of both mechanisms include inducing off-target effects because they are not as specific, and these trials have not been tested at a human level yet. Lastly, repeated administration is needed, which may be unappealing for many patients. Overall, researchers have hypothesized that targeting TDP43 aggregation using multiple therapeutic strategies, gene therapies, ASOs, small molecules, and protein drugs could reduce symptoms of ALS and extend the lifespan of patients. These strategies developed so far have been analyzed in this presentation, and overall, each has been effective in the animal models with specific criteria. But the most important limitation to many of these therapeutic strategies was that they targeted patients with known genetic mutations. So it cannot be determined how SALS can be targeted yet unless the mislocalization of TDP43 aggregation can be targeted. Since ALS is a debilitating disease that still has no effective treatment, any therapeutic pathway that extends the lifespan of patients would be a huge advancement in this field. Thank you.